Well, let's get started. So I thought the first chapter I've read in Janeway, I thought it was fairly easy to read, but long. So I learned my lesson to start a little bit early in the week reading. But it, I think, very comprehensively reviews the whole Peyer text in one chapter. So hopefully nothing too new, and just trying to drive home some of the major points and definitions at this point. Right, so the chapter starts by introducing immunology as the study of the body's defense against infection, and I actually broadened that into disease, because we know it fights cancer and autoimmune disease as well. A gentleman named Edward Jenner is credited as starting the field when he successfully created a vaccine against smallpox by inoculating folks with cowpox. He's made an observation that milkmaids who are exposed to cowpox don't develop smallpox. This is a picture of Edward Jenner, as you can see in page 1 of the text. So this eventually led to certainly a more refined vaccine for smallpox, and became the first disease eradicated on Earth, or thought to be eradicated on Earth. The last known case was in 1977. This gentleman here was the last known person to have smallpox, and he survived. 3 years after the announcement was made that smallpox was officially eradicated. There are a few very, very controversial files known to exist. I think one's in Russia and one is in the U.S., and they've tried on several occasions to destroy them, but it's never happened. And then I think just this summer, the NIH discovered they had some smallpox in a vial that was not known. So I think there are 3 known vials now, 2 in the U.S. So some other immunologists that kind of fathered this area, Robert, I don't know if it's Koch or Koch or Koch, proved that infectious diseases are caused by microorganisms. So the idea of contagion is very old, but it's actually a very, very modern idea that microorganisms are what spread infectious diseases. Then Louis Pasteur came along, and he actually made a vaccine against cholera in chickens. He also made a rabies vaccine and successfully saved an individual with rabies. And then von Behring and Kitasato discovered that, observed that the serum from animals immune to diseases such as diphtheria or tetanus actually contained antitoxic activity. So we know now that this antitoxic activity was antibodies that they were observing. Okay, so several definitions to get started. So pathogens, we know, are these disease-causing microorganisms. And then the antibodies are what is responsible for the antitoxic activity of serum aimed against the pathogens. The antigens are the actual targets on the pathogens that antibodies are targeting. And of course, the immune response, we very broadly break down into 2 categories, the adaptive immune response and the innate immune response. So the adaptive immune response has very specific immune responses, has memory, as opposed to the innate immune response, which is immediately available. It works very broadly, but has no memory and no specificity. Right, so the hallmark of the immune system is that it has 4 main functions. The first is recognition of the infection, so the infection can be detected. And then there are numerous effector functions, which we'll spend the bulk of this book reading about, that contain and eliminate the infection. But as we are increasingly recognizing over the past several years, is that regulation is of the utmost importance in the immune system. This is the body's ability to maintain control of the immune system so you don't develop autoimmune diseases or allergic diseases. And then, of course, memory, which leads to a more immediate and stronger response on second exposure to the same pathogen. So what is the immune system made of? Well, certainly many cells, which we call leukocytes or white blood cells, that originate in the bone marrow and are derived from hematopoietic stem cells. So this is a picture from page 5 of the text that starts with the pluripotent hematopoietic stem cell, and then immediately branches into either the common lymphoid progenitor or the common myeloid progenitor. So the common lymphoid progenitor, of course, makes the lymphocytes, which are the B, T, and NK cells. The B and T cells are the hallmarks of the adaptive immune response. NK cells are part of the innate immune response. And then the common myeloid progenitor is responsible for the other white blood cells, including the granulocytes, which are the cells that are very granular in nature, the neutrophils, eosinophils, and basophils, as well as the megakaryocyte and erythroblasts, which make up the platelets and the erythrocytes, and the monocytes, which terminally differentiate into macrophages in the periphery, in the tissue. And then mast cells, we know, come from the common myeloid progenitor, but we don't exactly know what the intermediate cell is. The dendritic cells are a little bit different. They actually bridge the gap between the innate and adaptive immune system, and we're going to talk a little bit more about this as we go on. So a little bit closer look at these cells. This is on page 6, a figure on page 6. So macrophages, of course, are one of our primary phagocytes. They often activate the immune response. They're a sentinel. They sit in the periphery and wait for a pathogen to get through. Neutrophils are a granulocyte and are other major phagocyte that can engulf and digest bacterial invaders. Dendritic cells, like I mentioned, they're in the periphery. They are a second sentinel, so they're always there, and they bridge the gap to the adaptive immune response. So they in themselves are part of the innate immune system in that they recognize via pattern recognition receptors, they engulf, and they travel through the lymph to the lymph nodes where they can activate the adaptive immune response via the T cells and the B cells. The final sentinel is the mast cell. These sit in the periphery, and these contain histamine, all these granules that are sitting in the cytoplasm that you can see. Prepackaged, the hallmark of mast cells is the reaction happens quick. As soon as the cell decides to start work, and well, nothing needs to be transmitted to the nucleus to get an initial response, everything just spits out, and you have a very quick response. Within 15 minutes, we'll see a mast cell response. And then finally, something that, of course, helps the mast cell, that histamine is very good at breaking down the thick skin of parasites, and then the eosinophil, which is housed in the blood, can come in and help with those parasitic infections as well. And then the basophil is kind of a blood version of the mast cell, promotes the allergic response and in a parasitic immunity in the blood. I think these are just individual. I didn't go through them all. Right, so the lymphocyte is the last one to talk about. These are very small cells with a very large nucleus. So the production of new effector mechanisms is very important in lymphocytes. They receive signals and they start working many hours to days later when then they can start making cytokines and differentiate and become effective cells themselves. NK cells is the last lymphocyte, but it's actually part of the innate immune system. It doesn't adapt. It doesn't have DNA that changes, but it will release granules that kill viral infected cells similar to CD8 T cells. So the cells live either in the tissue, in the circulatory system, or in the lymph. And of course, the lymphoid organs are broken down into either the primary or secondary lymphoid organs where these cells are actually made or where their effector mechanisms occur or are at least signaled to start their mechanism. So the primary lymphoid organs are the bone marrow and thymus. The bone marrow, of course, makes the B cells. The thymus makes T cells. If you had no bone marrow, you would have no B cells. If you had no thymus, like in complete DeGeorge, you'd have no T cells at all. And then, of course, these cells come to work in the lymph nodes, the spleen, and mucosal lymphoid tissue, the secondary lymphoid organs, which we'll expand upon in a minute. So the lymph is a very complex network. There's lymph in every tissue, and it will drain the tissue up through relevant lymph nodes, and then it will exit lymph nodes via other afferent lymphoid vessels. But this will all eventually come into the thoracic duct. And once they've emptied into the thoracic duct, the thoracic duct empties into this left subclavian vein. So eventually anything that goes through a lymphoid tissue can get back into the circulation. Hopefully not pathogens by then, but hopefully T's and B cells can be recirculated, find a new spot to work in. So the immune system starts with an inflammatory response, which we know consists of calor, dolor, rubor, and tumor, heat, pain, redness, and swelling. This is caused by cytokines, chemokines, and complement parts of the both the innate and adaptive immune response. So typically what will happen is you'll have a cell that the bacteria is recognized by, and it will trigger the cell to secrete cytokines and chemokines, which will bring other cells to the point of infection. So in this case, they're showing a macrophage which gets triggered by a bacteria. Of course, it's recognizing a pattern on that bacteria with a pattern recognition receptor. It's able to secrete cytokines and chemokines, and then it causes vasodilation of the vessels around the site of infection, leading to things like neutrophils being able to slip out through the endothelium and actually get to the site of infection and do its job as well. So recognition is one of the most important parts of the immune system, of course the initial part of the immune system, and is essential for the adaptive immune response to work. So typically what happens is that a pattern that is found on pathogens but is not found on self, that we term pathogen-associated molecular patterns, or PAMPs, is recognized by specialized receptors like pattern recognition receptors that are found on our cells, including antigen-presenting cells. And this has to happen to secrete cytokines to allow the immune response to occur. So if a T cell or B cell recognizes an invader that has not set off the immune response through other mechanisms, an immune reaction will not occur. We know this because we have to add adjuvants to our vaccines to induce inflammation to aid with the response. So we can't put in just an antigen alone. We put in something that is definitely going to be recognized on pattern recognition receptors, or at least start a cytokine reaction, so that cytokine milieu is in the correct phase to allow for a more broad immune response. So this is a picture. Page 12, it shows the macrophage and some of the different pattern recognition receptors, each of which we'll learn about as time goes on. Some of the common ones we talk about are toll-like receptors, mannose receptors, glucan receptors, scavenger receptors. We'll learn about all these, but they recognize patterns that are unique to pathogens that aren't found on cells. So after recognition, what happens? Well, an antigen-presenting cell, such as the dendritic cell, has to present that antigen to start the adaptive immune response. So where does this happen? Of course, this happens in the secondary lymphoid tissue. So the dendritic cell has traveled through the afferent lymph to the lymph node, where it is going to present on MHC along with co-stimulatory molecules to T cells, typically, which will then present to B cells, and both can get started and divide very quickly into clones. So each T cell has one set of receptors that is unique to a specific antigen, and it will divide and make a clone of itself so that receptor is the same and specific to that same antigen. All the different clones together make up what is called your lymphocyte repertoire. So all the different lymphocyte specificities that you have in your body is your lymphocyte repertoire. And of course, once these clones are made, they're all activated to help in this initial immune response. Luckily, there are mechanisms that will help recognize when a lymphocyte recognizes itself, and that lymphocyte will die by apoptosis instead of living to fight itself most of the time. Right, so what happens here is a dendritic cell in the peripheral tissue. It's engulfing a bacteria or other pathogen. It's jumping into the lymph and draining via afferent lymph to the regional lymph nodes, where it's going to go to the T cell zone and present an MHC to the T cells. It's going to activate them as the first step of the response. Once this happens, once a T cell recognizes it, well, you want lots of these T cells because you want it to go back and to fight infection. So it's going to start dividing. And some of these are going to be more specific for the antigen. Some of these are going to be slightly more, slightly less specific for the antigen. Ones that recognize self are going to die by apoptosis, but then you're going to have a pool of mature lymphocytes that are going to be able to eliminate that antigen because they are specific for it. Right, so this leads to the clonal selection hypothesis, which has 4 postulates. That one, each lymphocyte bears a single type of receptor with a unique specificity. Each lymphocyte is not going to have 2 different receptors, just one. 2, the interaction between a foreign molecule and a lymphocyte receptor is capable of binding that molecule with high affinity and leads to lymphocyte activation. 3, the differentiated effector cells derived from an activated lymphocyte will bear receptors of identical specificity to those of the parent cell from which the lymphocyte was derived. And then the last one is that lymphocytes bearing receptors specific for ubiquitous self molecules are deleted at an early stage in lymphoid cell development and are therefore absent from the repertoire of mature lymphocytes. So how does this recognition happen? Well, it's different on B cells and T cells. So a B cell essentially has an antibody that is membrane bound, and this is its B cell receptor. It is made up of 2 heavy chains and 2 light chains that together make up a variable region. 2 variable regions are antigen binding sites that are specific for an antigen, and then a constant region that's responsible for the effector function of that molecule. And then T cell receptors are made of an alpha and beta chain that together make up a variable region in a constant region, similar to the B cell receptors. So moving on to page 16, we see a figure of antibody. In this case, it looks like secreted antibody. That we can see the variable region recognized as something called an epitope, which is part of the antigen, and it is the part of the antigen that is recognized by antibody. Now, not every antibody has to recognize the same epitope. There's going to be several epitopes on that antigen that can be recognized by different specificities in antibodies. T cell receptors, however, recognize antigens in different forms. So the epitopes of T cells for T cell recognition are not necessarily on the outside readily available. So these antigens are first broken down inside a cell, and we'll talk much more about this as time goes on. And then these peptides are loaded on MHC to the cell surface, and then the T cell receptor recognizes the whole complex of the MHC molecule and the peptide together. It's not capable of recognizing it in a free form like antibodies are. All right, so a little bit more in depth about the lymphoid tissue. So the afferent lymphatic vessels are what brings lymph containing bits of pathogen and dendritic cells and other lymphoid cells to the lymph node. There are also arterioles that at the very end contain something called high endothelial venules, which have lots of holes in them that allow cells such as lymphocytes, T cells, and B cells to squeeze out to this particular lymphoid tissue. And then, of course, there's B cells, which are found in the follicle of the lymphoid tissue, which are in the outer cortex, and then when activated form germinal centers. And then, of course, the T cells are found in what I usually call the T cell zone, but are also known as the paracortical areas. So this entire circuit we've looked at before, but when you have an infection, the pathogen and the dendritic cell are able to travel to the lymph node and then recirculate through efferent to the thoracic duct and eventually back into circulation. Circulation is also what brings the T and B cells to the lymph node. So this is a nice picture of the lymph node that's on page 19, where you have your afferent lymphoid vessels, your outer cortical region where you have your B cells, and then these are germinal centers, so these are activated parts. The paracortical region is mostly where the T cells are found, and then everything is going to drain into a medullary sinus where it's able to exit through the efferent lymph. And of course, there's an artery and vein that will bring in B and T cells through high endothelial venules. This is one more picture, electron microscope of a lymph node, where you can see actually several germinal centers. The spleen, of course, is another form of secondary lymphoid tissue. It's primarily made of red pulp, which is important in red blood cell disposal. The white pulp, as the name would imply, is where the lymphocytes are. So these typically surround the arterioles. The T cells are found in a periarterial lymphoid sheath, or PALs, and the follicles are just like in the lymph nodes where the B cells are. In the spleen, there's a specialized kind of B cell called marginal zone B cells, which we'll talk more about as time goes on. So this is a nice picture of the spleen that shows mostly red, the red pulp, and then I think these actually are supposed to represent the white pulp around arterioles. But I think this is a better picture here that you can see the white pulp. You can see both the let's see, the arterial, and then you have your periarterial lymphoid sheath, so where the T cell zones are, and then the B cells, marginal zone B cells, which also are going to form germinal centers when they're dividing, when it's fighting against an antigen. This is just a slide of the same thing going on. So you have your arterial, which you can't see here, and then you have your periarterial lymphoid sheath where the T cell zones are, and then you have a germinal center forming here, marginal zones where the B cells are. So another form of secondary lymphoid tissue is the MALT, or mucosa-associated lymphoid tissue. Different examples include the GALT, or gut-associated lymphoid tissue, which includes the tonsils, adenoids, appendix, and Peyer's patches. The NALT, which is nasal-associated lymphoid tissue, and the BALT, bronchus-associated lymphoid tissue. So this is a picture of the Peyer's patch from page 21. So these are very similar to any other type of secondary lymphoid tissue where you have T cell zones that is more diffuse, and then the better defined follicles, which contain germinal centers when there's active B cell proliferation. So you have your same afferent efferent vessels, which are going to drop the T's and B's into the Peyer's patches. But you don't have afferent lymph in this case. All you have is an M cell, which is going to pinocytose things from the lumen of the gut here and bring it into the germinal center, where the same reaction can happen. It'll be recognized by a T cell, and everything still drains via afferent lymph, eventually to the thoracic duct, into the left subclavian vein, back into circulation. So what happens when these lymphocytes are getting activated? Well, like all things in the immune system, there has to be a primary and secondary signal. So two things have to be happening, and this is just a fail-safe to make sure that your immune system isn't running amok and trying to fight off even self-antigen. So just like you need an adjuvant to cause inflammation to cause the response, well, when the lymphocyte is getting activated and it's getting presented an antigen, well, it needs a second signal, typically a co-stimulatory molecule. So if there's no co-stimulatory signal at the time antigen is presented, then it may lead to anergy. Or there may be something that's actually competing against the co-stimulatory molecule, which is going to lead to anergy. So this is from the bottom of page 22, and it shows a dendritic cell, and it's just trying to drive home the point that the dendritic cell is going to present to the T cell on the T cell receptor, but it's also going to be giving it other signals, and not just one, probably several signals to direct this T cell to become activated, and in what way to become activated, which way to differentiate, and what to do. Similarly, the T cell then, of course, is going to activate the B cell. We know that the T cell has to be activated first for a B cell to be activated. Well, it's going to be receiving more than one signal, too. It's going to be receiving a signal through the B cell receptor from the T cell, but it's also going to be directly recognizing and binding to antigen. So the different things that can present to T's and B cells are antigen-presenting cells, dendritic cells, macrophages, and B cells, or B lymphocytes, are all examples of antigen-presenting cells. These show different depictions of dendritic cells, macrophages, and B cells. And after activation, the lymphocytes are unique that they will have memory. So there's a primary immunization that takes place, which is like what we've talked about when the dendritic cell presents to the T cell in the lymph node, which eventually activates the B cell and then becomes effector cells, travels back to the site of infection or to the bone marrow. But this takes days to accomplish. So when you're sick, you're probably going to be sick for several days until the pathogen can be destroyed in the body. However, the second time, the secondary immunization, booster immunization, or the second time you are in contact with this, you probably won't have symptoms at all because there's memory, and the T cells and B cells will be able to act even more quickly to fight that infection. Something does happen only in B cells and not to T cells, that as time goes on, the B cells can actually mature and have a higher affinity and even stronger response to that specific antigen. From page 24, this is a picture showing the difference between the primary response and the secondary response. So it takes several days for the primary response and eradication of the pathogen, but then the second time around, it's a very quick, very strong, may not even feel symptoms. Okay, on to the second part of this chapter where it talks more specifically about the effector mechanisms of both B cells, which is by antibody, and T cells, which are differentiated into CD8 and CD4 cells, which have different mechanisms. So the reason we have different types of adaptive immune responses is because they need to fight different things. So the main types of broad categories of pathogens are extracellular bacteria, parasites and fungi, intracellular bacteria, viruses, and parasitic worms, that all need different mechanisms to fight. So examples of extracellular bacteria, parasites, fungi, strep pneumo, clostridium, pneumocystis, intracellular bacteria such as mycobacteria, viruses such as the flu that we're all trying to fight right now, and parasitic infections, including schistosoma. Right, so the B cells are the humoral part of the immune system. So after the broad breakdown of the innate and adaptive immune response, the adaptive immune response can be further divided into the humoral and cell-mediated response. So the humoral, or B cells, happens via antibodies, and there are several different antibody classes or isotopes that all have different jobs. But broadly, antibodies have three mechanisms in that they neutralize, opsonize, and activate complement to fight infection. So here's a nice picture from the text on page 27. So for example, if there is a bacterial toxin, it can be quickly neutralized by antibody, so it can block it from interacting with its target or its receptor. But then once it is neutralized, it can be ingested by a macrophage and destroyed. It also, excuse me, can help opsonization. So bacteria that is not inside the cell, so extracellular bacteria, will be bound by antibody and make it actually easier for ingestion by the macrophage. And it can also activate complement, particularly IgM. So it will get coated, and this will start the complement system to make MAC complexes on the outer surface and cause lysis and eventual ingestion and destruction. The cell-mediated immune response, or the T cell side, is broken down even further into cytotoxic T cells and helper T cells. Cytotoxic T cells are identified by the CD8 complex on them, which is a co-receptor for the MHC. Of course, it recognizes MHC class 1, both the T cell receptor and CD8. The helper T cell, or CD4, has a CD4 co-receptor that recognizes MHC class 2. The helper T cells can be further broken down into subtypes, including Th1, Th2, Th17, these follicular helper T cells, which weren't talked about in the box text much, so this is going to be a new one for me too. But we know many more subtypes now, like Th33, all of which have their specific job in the immune system. So the T cells are primarily battling, like the antibodies, we're battling extracellular bacteria, so things on the outside of the cell. T cells are fighting intracellular bacteria, so this can happen in two ways. So if you have a viral infected cell, well, a cytotoxic T cell is going to be able to sense that on the outside and will signal the infected cell to die by apoptosis. Or there's a helper T cell that actually gives signals to the cell to help it increase its killing ability and engulfing ability. So it, in the case of, say, Mycobacterium here, where there's an intracellular pathogen, it ramps up all the mechanisms that help this macrophage destroy the invader. So in the cell-mediated world, this all happens via signaling through MHC class 1, or presentation from MHC class 1 and class 2 receptors. So we'll probably have a whole chapter about how a peptide is loaded onto either MHC class 1 or MHC class 2. It travels to the surface, and then T cells can only recognize peptide or antigen, the pathogen, when it is bound inside of an MHC. So these are prepackaged after they're already engulfed or intracellular. The pathogen is engulfed or intracellular. It's formed in one of a couple different ways into smaller packages. It's cut up and then put on MHC, where it's then brought to the surface and can be recognized by a T cell. So this happens via both the T cell receptor recognizing the MHC, but also the co-receptor, CD8 or CD4, recognizing the same MHC molecule. This is not a co-stimulatory molecule because it's recognizing the same thing that the receptor does. The co-stimulatory molecule, it would be recognizing a completely separate receptor here on the antigen presenting cell. This is just one more. This was actually a CD8, exemplifying the CD8 interaction. It's no different for CD4. It's the same concept. It's a co-receptor. It also recognizes MHC as opposed to its own receptor, but then it helps B cells, activate B cells. Can also help activate macrophages or other neutrophils, phagocytes in the periphery. And that's pretty much it. I didn't cover in this lecture too much about the disease states, including allergy, cancer, autoimmunity, immune deficiency, all of which are important and we'll learn about in due time as the year goes on. But it does have one summary slide at the end on page 35 that I think is very nice, that nicely wraps up the important points, including the innate immune response, which is important to start inflammation, which is necessary for the rest of the immune response to occur. It includes complement, phagocytes, NK cells, works very quickly, as opposed to the adaptive immune response. T cells, B cells takes several days for its effector response to work and eliminate the pathogen. But the adaptive immune response, unlike the innate immune response, can lead to memory, which can be lifelong and will cause a very quick and robust second response when you see a pathogen again. All right, that's it for today. Next week I don't think we have immunology, but the following week we will cover Chapter 2, which is more in depth about the innate immune system. I think it's a fairly long chapter. Let's see. Well, another 35 pages.
Yeah.
But I think this maybe leads a little bit, but this is going to be, Chapter 2 is going to be the first time this year that it's going to be very, very memorization fact heavy. So first years don't get bogged down, just try and get through it. Second year is the time to start concentrating on some of the details and maybe making note cards as you go along. There is a very nice website that goes with this book that I started to look at, but it actually has some flashcards to review some of this that maybe, even if you don't start going through them, at least organize or kind of get your system together. Another idea is to start your own PowerPoint and then pull down my PowerPoints will stay, so you can either pull the pictures down from the website, or if you go into the Allergy folder, Immunology 2014-2015, my lectures are there, and you can pull ones that are going to start having a lot of detail to memorize. Let's see if I can find an example quickly.
The electronic version on our iPads, you can make note cards directly off pages.
Oh, nice.
Oh, okay. That's nice. I was trying to figure out how to make notes on here, and I was not successful, but I think we're playing a little bit more.
Oh, so like on page 50 in Chapter 2 would be an example of maybe a picture you'd want to put into its own PowerPoint of all things before the board that you're just going to sit down and memorize very quickly.